today we're going to start talking about plant hormones and plant responses. So these are what's called the big five. So these are the plant hormones that we're going to talk about. And today I'm going to start with auxin. So auxin is something that you came across at GCSE, and it affects the direction that the plant will grow towards. So if we have a look in a shoot, so in the very tip of a shoot, you have cells that are receptive to light. So they will detect the direction of the light. But just behind that, you have some cells that produce auxin. Now what auxin does is if you have a plant cell, so you've got a cell wall and then a cell membrane, what auxin does is it loosens up that cell wall a little bit. So it makes it a little bit more elastic, there's a little bit more give in it. And so if water enters that plant cell is able to expand. You've given it a little bit of expansion room so it can get a bit longer. So what happens is these cells detect the direction of light. Cells just behind the tip are where the auxin is produced. Now, if the light is coming from directly above, then these cells are producing auxin, and what happens is that auxin will diffuse evenly downwards, back, kind of just in the direction of gravity. And so all of these cells will evenly be able to elongate, and when they elongate, that's going to help push that plant upwards. They're going to get longer, stretch upwards, and so your plant is going to be moving in that direction. So what about if the light is coming from the side? So if the light is coming from the side, then again, these light receptors are detecting the light. The cells just behind are producing the auxin, but what happens is the auxin will diffuse away from the direction of the light. So you'll get more auxin going down the dark side. So you may still get a bit of auxin going down the rest of the shoot, but most of it will diffuse across and you'll get more of it going down this dark side. So what happens here is that these cells are able to elongate. These cells don't elongate as much, and so that's what pushes that over. So if you think about one of those kind of bendy straws, if you bend it over, you'll notice that on that outside edge, it's actually longer than that inside edge. And so if they are longer, it's going to push that side to be longer and push the coleoptile over. Now obviously that's a good thing for the plant. It wants to grow towards the source of that light. That's how it's going to be able to produce its food. Now in the root it has the opposite effect. So when you've got a seed and your first root starts to grow out, the auxin is still going to be produced in the tip. It's going to start to diffuse backwards, but then it's going to fall and collect on the bottom. It's going to fall just due to gravity. It's going to start to collect on this lower side, and when it does that, it inhibits elongation. So these cells are going to continue to grow, those ones not so much, and that's what pushes that downwards. Helps to anchor the plant. Okay? So that's auxin in the tip and in the root. And next I'm going to talk about gibberellin. Now gibberellin is useful for two different things. It stimulates elongation in the internode and seed germination. Now, what happens with the elongation is that if you kind of draw a plant and you get some leaves, it's a terrible drawing of a plant, then this area between the nodes, the internode, will get longer. And there are some very nice pictures online of cabbages that are kind of 7 foot tall, 8 foot tall. Now, the way that they discovered that was the effect of gibberellin was because they noticed there were dwarf plants, and there was a cascade, it was a sequence of events with the formation of long and complicated pathway, and you finally end up with your gibberellin at the end. Now, what they discovered was there were two different plants that they were looking at. One of them, for example, couldn't do the top half, the other one couldn't do that last step. So one of them was missing an enzyme, and one of them was missing an intermediary. But either way, neither one of those plants on their own could do the whole pathway. So what they did was they grafted these two plants together. They took the top half of one and the bottom half of another, stuck them together. And when you do that, you then have that complete pathway. And so those dwarf plants could then grow tall. So essentially, if you've got this plant that's dwarf because it doesn't have one part of the pathway, and this plant that is dwarf because it doesn't have the other part of the pathway, if you kind of cut and stick the two together, what that one missed, this one now has, and what that one missed, this one now has, and so your plant will actually now be able to grow tall. Okay? Right, and so then the next bit with gibberellin is seed germination. Now, in seeds, you've got a store. So inside your seed, you've got starch. And your starch is a store of energy. Now that energy, that store, that starch can be broken down into glucose, and then that glucose can be respired. Now, your seed is like a little self-contained package. Until that plant has leaves and can photosynthesize to make glucose, it's got to survive on its own. So that is its start place, its store of energy before it gets some leaves. So what happens is water enters the seed, and water stimulates gibberellin. Now, gibberellin will actually switch on the transcription of the amylase gene. Now, amylase is the enzyme that breaks down starch. So water enters the seed, activates gibberellin. Gibberellin switches on the making of this enzyme, and that enzyme is what breaks your starch down into glucose. So when water enters and gibberellin is activated, the seed can access that stored energy. It gets broken down into glucose, and it can use the glucose to grow. So it will send out that first shoot, it will send out that first root. This shoot will then start getting leaves, and then the plant can photosynthesize and